Hello, I'm Dr. Martin Moreed, and I'm excited to share with you today some research that we're just publishing in the Journal of Biological Rhythms. My team at CIRCADIAN has identified for the first time a narrow blue band of wavelengths, so-called circadian blue, which synchronizes our circadian clocks, and when incorporated into lights, enables us to deliver lighting that's safe for today's world. Now, blue light is already well established, and you're probably already aware, as a public health hazard when we're exposed to it at night. Any type of blue-rich lighting, whether it's LEDs or fluorescence during the nighttime hours, is associated with obesity and diabetes, sleep disorders, mood disorders, and even certain types of cancer, such as breast cancer and prostate cancer. But blue light is not always the villain. It's also the hero too, because during the daytime hours, blue light is critical to ensuring our circadian rhythms are synchronized. It enables us to sleep well at night. And also, it enables our immune system to be as strong as it can be, which is so critical in today's world. However, there was a fundamental question that had not been answered about blue light. What are the precise wavelengths that we need to control to ensure that we have lighting that is healthy and safe? Now, that simple question has actually had a lot of confusion around it. There are different models out there: circadian stimulus model, equivalent melanopic lux model, the CIA 2018 standard, all talking about different versions of what the spectrum is that needs to be controlled. And to add to the confusion, various manufacturers are presenting products which are said to control blue with defining different ranges of spectrum of blue in their particular model features. So all that was a matter of such confusion that we decided we better delve into this and figure out what was going on. And what we discovered was rather interesting. What we discovered is the core research that all these models were based on was research that was done in the dark. This was with dark-adapted subjects or dark-adapted isolated retina or even the melanopsin pigment, all studied in the dark and then exposed to short monochromatic, that's single-color lights, for short periods of time to look at the response. None of it was done in the conditions of the real world. And that is what we've set out to do and what we're able to report in this paper. So what was vitally needed was to measure the circadian response under conditions that are normal to the workplace or school or home, and do it with regular polychromatic white light, normal white light, do it with light intensities that were typical from anything from 200 lux to 1000 lux, to look at people who are freely moving and not restrained, to look at them being exposed to light for the normal lengths of time, 8 or 12 or more hours at a time, and above all, to use a measurement that was health-related. And the key measurement we picked was the overall total production of melatonin over the course of a night, because that is highly correlated to the risk of breast cancer, diabetes, prostate cancer, mood disorders, a whole variety of problems that are result of circadian disruption. In our studies, we used a very interesting feature of white light. It's made of a rainbow mix of colors, but you can achieve white light with very different combinations of those colors. That enabled us to select 7 different light fixtures. Some were LEDs, some were fluorescents, and we were able to have them all light the same conference room, provide the same level of lighting, which was 50-foot candles, 540 lux, normal conference room lighting levels, and then bring in the National Institutes of Health grant, a total of 34 people under the different lighting conditions. They were both men and women. They were the same people being exposed to different lights, but under the same light intensity. And we found some very interesting findings. We found some of those lights were highly suppressive of melatonin, whereas other lights allowed melatonin to rise to its normal levels. That enabled us to dissect out what was in fact the key wavelengths that the system is responding to, the circadian system is responding to in regular light-adapted conditions. So when we analyzed all these different light spectra, and the patterns of melatonin each of them produced, we were able to tease out and identify for the first time the color of circadian blue light. Circadian blue light has a peak wavelength at 477 nanometers, and it's a relatively narrow, sharp peak, so the half maximum effects are obtained at 438 to 493 nanometers in the blue. It has very little in the way of violet or green components to it. Now that we've identified circadian blue, the precise wavelengths of light that stimulate the circadian system, we have the key to unlocking the door to circadian lighting. We can build LED chips, light fixtures, light bulbs, which automatically, during the daytime hours, strengthen our circadian rhythms, providing circadian blue-rich light. And during the evening and night, protect us from the harmful effects of light at night by removing the circadian blue stimulus. What we've done, therefore, is pave the way to engineering light for human health and well-being.